All right. Thanks for coming, guys. So today I'm going to just finish up the previous lecture on modeling metabolism. So we're going to talk about the Gillespie algorithm, which I think is super important and like very useful in many situations above and beyond those that we're going to talk about. And then I want to give you guys a sense of like what the interesting problems are in metabolomics. So personally, like my research has always been on microbiome, and I think that's pretty exciting. But in terms of being like immediately practical and useful, let's say as a diagnostic for human health, it's okay. What I've noticed is if you can find an association between a disease and the microbiome, you can get an exciting paper, and a lot of people will read it and be impressed by that. But when you actually compare the quality of diagnoses and predictions that you can make from, let's say, microbiome compared to something like serum metabolomics, there's like the metabolomics is much more impressive. But if you find a diagnostic that's based on serum metabolomics, usually don't get like as impressive of a paper. People aren't that impressed because like obviously if you look at like kind of the sum total of all the metabolites in the blood, it tells you a lot about that person's health. Um, and there's a number of reasons I think why metabolomics hasn't really taken off as much as some other fields in terms of like being this kind of catch-all general diagnostic. And part of it is the fact that many of the other technologies we have, like genome sequencing, we went through this phase where we took a hit in quality to massively increase the throughput. And in general, people doing analytical chemistry, like they want machines that are even more accurate. And, you know, even if there's even at the expense of bandwidth. But I think that's starting to change. And so I think for you guys, this is a super exciting field to get into. So I'm gonna show you some cool algorithms, especially one that's being developed by Connor Coley's lab here at MIT. But the thing I kind of want to impress on you, I'm going to give you the basics of interpreting mass spec data. I went over to the Broad Institute last week and I said, hey, I'm giving a lecture on kind of the state of the art in machine learning and computation and metabolomics. Sat down with the director of the Metabolomics platform, and he kind of told me like, look, a lot of the algorithms we're using were developed decades ago, and like the real advances in the field are going to come from some of the really basic stuff, like peak calling and aligning peaks across experiments and things like that. So um, today I'm not going to give you this polished thing like AlphaFold 3 and say, look at all the great things that other people are doing. I'm going to tell you guys about the basics and what the problems are. And I think this is like a super rich field. You know, if you're thinking about like, oh, where do I want to apply machine learning in the future? Where is there a bunch of big data that's kind of waiting to be turned into exciting machine learning or AI based models? Okay. Before we start on that, though, I want to go back to stochastic modeling. So last time we looked at analyzing the stoichiometry matrix for a genome scale model, and we saw that because of the way the biological pathways are set up, you often have a choice of processing mass through more than one different pathway. So you have this underdetermined system, and you can figure out what an optimal solution is if you know what's being maximized, right? Like biomass or ATP production or NADH. If we want to look at kind of zoom in on particular pathways, and maybe even look at like what are the dynamics going on in a single cell, sometimes we're talking about small number of molecules, right? So if we take a flux balance approach, now we're assuming that the number of molecules doesn't change, right? If we take a ODE type approach, now we're assuming it changes, but we're assuming it behaves like an ordinary differential equation, and that's only true in the limit where we have many, many molecules. So oftentimes we're looking inside a single cell, and we want to know what's going on here. And we can think about this in terms of metabolism and small numbers of metabolites being created and then used as substrates for other reactions. But we can also take the same modeling approach and apply it to things like transcription and translation and these other processes that happen in the cell where, if you're transcribing a single gene, maybe there's only like one RNA currently in the cell, or maybe there's like 5 or 10. There's these small molecules, and so then the fluctuations you get are significant compared to the total amount of that transcript. So in those cases, there's a really nice algorithm that I think is intuitively very elegant for modeling those things. And it's called the Gillespie algorithm. So the idea behind Gillespie is we're going to do essentially 3 things. First, we're going to calculate something that they call the reaction propensities. Okay. And so a reaction propensity is sort of like given the current state of the cell, and that includes, how many of, let's say, each of the substrate molecules there are in the cell. What is the rate of the reaction? Okay, so if the rate of the reaction is, let's say, 10 reactions per second, but then there's another reaction that we're modeling, and the rate of that reaction is 5 reactions per second, then each of those rates becomes a reaction propensity. And so we can add up all of those propensities. So if we only had these 2 reactions, it would be like 10 plus 5. So there is some reaction in the cell happening at a rate of 15 per second. We just don't know which one it is, right? That's the basic idea. So in the first step, we're going to calculate the reaction propensities. We're going to add them together, and we're going to find the overall rate of reaction. And then once we know the rate at which something is happening, right? In this case, there's 15 somethings happening per second. We're going to sample the time to the next reaction. We don't know what kind of reaction it is, but we know how long it's going to take to get there. And then the next step is going to be, okay, we know something happened. What's the probability that it was reaction 1 or reaction 2? And that's pretty simple because it's just, reaction 1 was 10 per second, reaction 2 is 5 per second. So it's twice as likely to be reaction 1 versus reaction 2, right? So we're going to essentially do the stochastic modeling by picking 2 random numbers. Okay. So our reaction propensity, we're going to calculate that based on the everything that's going on in the cell. So once we update our system, and like we've been running our reaction for a while, maybe initially we started out, in reaction 1, there were 0 molecules there to serve as substrate for the reaction. So now, even if it has a really fast rate constant, the propensity of the reaction, because we've got the rate constant times the concentration of each of the products, is going to be 0, right? And as we start to build more and more of that substrate, then the propensity and the rate of that forward reaction is going to go up. Okay, so we'll calculate the reaction propensity. We'll calculate the next reaction time, and then we'll store the state of the system in a big vector that says, how many molecules of each thing are currently present in the cell. Okay, so I kind of give you an overview of the general algorithm, but we can break it down more precisely. And so initially we'll set up the system with some initial concentrations. And then the first thing we're going to do, calculate all the reaction propensities. Okay, so all the rate constants times the the amount of each. Then we'll add those up to get this total propensity number. Okay, and then what we want to do is we want to figure out, what is the time that we have to wait until the next reaction, right? So if we have a reaction rate of, let's say, 1, total reaction rate of 1 every 10 seconds, then what is the waiting time until the next until the next reaction? That just ends up that's just basically like an exponential decay. Okay, so if we simulate a random number between 0 and 1, then if we take the natural log of 1 over that number, that's going to be that's going to be our waiting time. Okay, and we scale that, of course, by the total reaction propensity. Okay. All right, so that's just a way, and that formula is pretty straightforward. If we were to just look at our exponential decay and we made a cumulative distribution function, just measuring the probability that it hasn't happened yet, that's where we would get that equation. So now we know, by drawing from this distribution, we're just drawing from a uniform between 0 and 1. Now we've got the time to our next reaction. And then we just have to look at all the individual reactions, and we're going to weight them by how much, basically how fast their rate is, right? So if one was twice as fast as the other, then its probability will be twice as high. And so we just pick another random number between 0 and 1. We just line up our reactions in a row, and if it falls in the space, we allot space to the reactions based on how fast they are. And if our random number falls within a certain block, then we just say it's that reaction that happens next. So each step through the algorithm makes one molecule, so it's kind of slow. And that's the price we pay to really get a very rigorous handle on how this variation in the small numbers of molecules is going to affect the dynamics of the system. Of course, we're simulating one trajectory at a time. So in order to use that, we have to simulate many trajectories through the system. So that's when it does actually get pretty computationally expensive. So a lot of times you'll see the Gillespie algorithm used in sort of a hybrid method, where if we have a few species that actually, maybe there's thousands or millions of molecules or something like that, we don't want to include those in the stochastic part of the algorithm. So we can also simulate those using differential equations and just use Gillespie for the more lowly abundant species. Okay, so I think I mentioned this, but this is it's computationally expensive for large systems, especially because we usually want to get the average behavior of a system. These types of models were super interesting to me. So when I was in graduate school, I was doing protein folding work. And when I was thinking about, oh, what do I want to do like later in life? I found these models really compelling. And at the time, my postdoc advisor, like before I had joined his lab, he was building simulations of entire organisms. So he was focused on lambda, the E. coli lambda phage. And so he had programmed in all of the different components of that organism. So, basically modeling each ribosome individually and each RNA polymerase and each mRNA, and then using the Gillespie algorithm to simulate how the phage behaved. And so this is an example of the lambda phage genome. And so you can see there's not much going on to make a lambda phage, but a lambda phage has some properties. It has some internal logic, and it's been honed through evolution to maximize its own reproduction. And it does that, partly by making this decision of, when to integrate into the genome and then when to actually start copying itself and lyse the cell. So if it integrates into the genome, then it can stay in the genome for a while. And if conditions are good, that cell might replicate. And then there'll be more copies of the lambda phage as well. So it has to make this decision. And one of the cool things is it can make this decision in a way that's stochastic. So it can sort of pick a random number, and then, if conditions are right, 99% of the time it'll integrate into the genome. 1% of the time it will go the other way. And under different, slightly different conditions, it can flip and, mostly copy itself and lyse the cell, and only very rarely go into the genome. Okay, so it has to do all of this with a fairly small number of components, especially because most of these components are just part of the housekeeping machinery of the phage itself. So there's not a lot of room in here to encode a lot of logic. So the exciting thing that had happened was, people had built these diagrams of what the internal logic was based on how these different genes interacted with each other. And what my advisor, Adam Arkin, had done is he has a number of papers kind of analyzing the system. And you can see from different starting points on this landscape, and here the landscape is simplified. It's the amount of two different proteins in the cell that kind of, I think they basically repress each other. And one goes towards replicating the virus, the other goes towards integrating in the genome. You can get deterministic behavior if you have like an ODE type model where you always stay under certain conditions, you always stay in like one or the other. But then if you actually run the stochastic model, you can see that there's actually some randomness, and you can jump from one to the other. So it not only incorporates some randomness, but that noise also allows you to build more sensitive components. And so people have taken advantage of this to build different types of circuits in synthetic biology where they want to increase the sensitivity, let's say, of a certain detector. The noise can allow you to do that. And so these simulations are a way to start to model and understand that noise and how it works. Okay, so that concludes the modeling of metabolites. Now I want to switch to measuring metabolites. and give you guys kind of an overview. So, like I said, these metabolites, especially if you're measuring, let's say, circulating metabolites in blood, these could be really informative and helpful in diagnosing um, patients and just generally quite informative about the state of the organism. We can also identify particular compounds. So if you do an experiment, you know you've got a molecule maybe that increases inflammation. You think this might be important. You can use these techniques to try to identify those molecules. And most of what we'll talk about today is looking at a complex sample. Let's say, if I'm doing microbiology, I might be looking at the inside of a cell. I might be looking at its waste products. And that's always a choice when we're doing metabolomics, especially with looking, let's say, at the human microbiome. We want to know like what metabolites are important. So we have to make that decision. Okay, are we looking at what's outside the cell? Are we looking at what's inside the cell? So we can wash off those cells, spin them down, break them open. Are we looking at both? Like what, what are the metabolites that we think are important? And once we've made that decision, then generally we have a number of different metabolites, and so it's difficult to uniquely identify all of them. And so we sort of take this very general approach. Let's identify as many as we can, and then maybe come back if we find certain metabolites that seem to be important or changing under the experimental conditions that we care about. We can come back and use other sorts of techniques, but primarily we stick with mass spectrometry, and that's what I want to talk most about today. Okay. So apart from mass spec, which we'll get into, there's NMR spectroscopy. NMR is great, usually for systems that we've purified them a little bit, so we're down to like a smaller number of components, and we usually need a little bit higher quantities or higher concentration of those metabolites to use NMR. But it can be really informative about the identification of the compound and also quantitation. It can be very accurate. And then there's other techniques that are potentially easier and can give us some idea to help us make an ID from just the mass spec. Okay, so the rest of the day we'll talk about mass spectrometry specifically for small molecules, but I'll mention proteomics briefly, which is another really exciting use where there's also a lot of room for innovation on the computational and algorithmic side. But essentially in mass spec, we have a few steps. First, we need to take our sample, and we need to take the molecules that are there and ionize them into charged species, because that's basically what we're using to figure out their weight. So we can think of a mass spec as a really fancy scale. We're measuring the weight of things. We're measuring the weight one molecule at a time. And the way that we're doing that is by ionizing that molecule and then seeing how it behaves in an electric field. So we want to ionize them. We're going to separate them based on this ratio called M/Z. So it's the mass of the molecule over the charge, right? And so if we're accelerating them in an electric field and usually a magnetic field as well, the amount of acceleration that we see is going to depend on the mass and the charge, right? Because the charge is going to determine the force, and the mass is going to determine uh, the acceleration. So once we have those molecules, then we just need to detect them and count them. And there's a few different ways that we can generate charged molecules. One, if we just have maybe like one compound and we want to get an ID on it, we can use like hit it with an electron beam, and that will result in a certain fragmentation patterns, and we can use that to identify a compound. Usually, if we're doing metabolomics on complicated samples, we'll use something called electrospray ionization or other kind of softer types of ionization. And those tend not to fragment the molecules as much. So the main peaks that we get are related to the mass of the entire molecule. Whereas, in some of the harder ionization techniques, that molecule is going to break up into pieces, but the specific weights of those pieces are going to tell us a lot about the like what type of molecule it is. Okay. So once we have our ions, then we're going to put them through our mass analyzers. There's a number of different types of mass analyzers. Probably the most common is the quadrupole machines, but there's other ones that are worth knowing about. So here's kind of like a broad overview of quadrupole machines, time of flight, which basically is exactly what it sounds like. It just measures the time it takes for that ion to hit the detector. And then there's fancier machines like the Orbitrap or ion trap and the Fourier transform ICR. I don't remember what ICR stands for. But like the Orbitrap and the FTICR, they can measure masses down to like depending, like 5 or I think sometimes even like 6 decimal places. So what that means is instead of just getting a mass that's that tells you about the atomic weight sort of in like a integer number of Daltons, you can actually get what they call the mass defect. And so, when we have two atoms of carbon are, let's say 12 Daltons, but when they bind to each other, now their potential energy has gone down, right? And so the mass also decreases. Okay, by like ever so slightly, but if we can measure like 5 decimal places, then we can actually like see the mass defect for all these different types of atoms. And so when we get a mass that precise, instead of just knowing kind of like how many Daltons it is, we can actually figure out what the chemical formula is. Like we know how many carbons, how many nitrogens, how many hydrogens, things like that. Those are all pieces of information that then can go into later doing our analyses. Okay, so what's a workflow? We have to collect samples, prep those samples. Usually, if it's a complicated sample, we need to separate the metabolites, so we're not looking at all of them at once. Although you can kind of, even for a complex sample, you can take it and do methods like fingerprinting or direct injection. We put the whole sample in at once, and we get like a lot of peaks. And then part of the exciting, new things going on in the computational metabolomics are trying to decipher what what those experiments are, but fundamentally they're going to be limited. They're going to be limited compared to your ability to identify and quantitate compounds when you've actually separated them before they get to the detector. We have our detection, data processing, and then at the end of the day, the statistical analysis based on our experimental design. So for metabolomics, the sample prep kind of involves like two stages. First, we have to do something to stop the metabolic activity. Even if we kill the cell, that thing is full of enzymes, and a lot of those enzymes work like just fine in vitro. Also, depending on the pH and things like that, the redox potential, we can have a lot of reactions happening on the like internal components of the cell, even if even if the cell is not alive, even if there's no biology, there's chemistry that takes place. And so like the inside a living cell, things are always happening, whether they're biologically catalyzed or chemical. So we have to do something to stop it. That often involves, we can put them directly into liquid nitrogen, can put them into oftentimes like really strong acid conditions, as well as putting them into liquid nitrogen. Then we need to extract the metabolites that we care about. Number of different ways to do that. And then potentially derivatize them. And how we perform these sample preparation steps is going to impact our ability to compare across different experiments and across different laboratories. Okay, so most of what we'll talk about in terms of separation is chromatography and primarily liquid chromatography. And so in liquid chromatography, there's a number of different columns that can be used, and they generally either separate based on polarity or hydrophobicity. Similar concepts. And the idea is that, if I have a hydrophobic column, then that's going to retain, molecules longer based on how hydrophobic they are and maybe based on their size as well. But the point is really to take different molecules and try to separate them as much as possible from each other, so that as they come through the chromatography, we're taking little samples of that, and then we're putting it into the mass spec, right? So we're taking a mass spectrum, continuously as we're flowing our sample through the chromatography column. And so there's basically we're generating data on two different axes. So we're measuring intensity, which is essentially like how many molecules are hitting our detector, and we're scanning our sample coming through the chromatography column over and over again, and then we're looking at what's coming out of our mass analyzer. So like where is the intensity in terms of M over Z, this M over Z ratio, right? Which is telling us basically the mass of the compound. So we have this kind of two-dimensional data. Here I'm showing it to you in this really simple form. It's more complicated than that, and I think I mentioned at the beginning when I went over to the Broad and I was asking, hey, what are the big computational problems? One of the big problems is how do we reliably take the raw data coming out of the machine, and how do we identify these peaks that look so discrete here? Because if we could do that really well across different experiments, that would give us like a lot more power to analyze these samples. Okay. So there's a number of different tools that we can use for data processing, which we'll get into. Uh, quality control. So there's kind of two types of metabolomics that we can do. One is targeted and one is untargeted. Here I'm mostly talking about untargeted, where we're taking kind of everything that comes through. But in either case, there may be a few targets that we really care about. And it may be that, looks like Manoli's getting a call. Oh, I can't seem to end the call. All right. At any case, there may be a few specific targets that we care about, and if that's true, then we can kind of spike our sample with standards of that molecule. Usually, we'll spike in standards of the molecule that have unusual isotopes of carbon, or one of maybe nitrogen. And then we can identify where those standards are occurring in terms of the retention time, because those, the slightly different mass, it's probably not changing how it's moving through that, let's say, hydrophobicity column. And it will just be offset by a predictable amount from the M over Z of the actual compound. And then if we know additionally how much of that standard that we put in, then we can quantify, what the we can quantify how much of that compound is in our actual sample by taking a ratio between those two. In general, it is difficult. So mass spec is it's fairly quantitative, because we're literally counting, in many cases we're literally counting the number of molecules that we see. But for every and we'll talk about this later, like we are only seeing some of the ions, right? We might only be seeing like either only seeing the positive ions or only seeing the negative ions in any one experiment. And so what fraction of the total number of molecules in the sample are getting ionized, and what fraction of them are getting ionized in different forms. So some of our positive ions, they might be missing an electron. They might have an extra proton. They might have an extra sodium. There could be a lot of different forms that we see. And those different ionizations might happen in different amounts. And so it's because of all these uncertainties that this technique, which should be fairly accurate and fairly quantitative, becomes less accurate and less quantitative. So that can kind of affect things. And then if we don't have great separation in our chromatography, there's only a certain number of ions that we're going to see in any particular spectrum. So if we have two molecules entering at the same time, and one of them is very efficient and it's sucking up like all of the available ions, then the other one is going to be depressed, like artificially, and that's going to also throw off the quantitation. So it can be fairly tricky to try to quantitate these things. And that's why, we'll run replicates, internal standards, and also calibration curves to try to account for that. Okay, if you guys are going to be working with metabolomics data, you should be somewhat familiar with like how to get and use the data. So generally, when you get data off of any particular machine, it'll come in some format that's usually unique to the vendor of that particular machine. And then there's a number of different software tools that will read in those formats and then output them in an open format. So a lot of people use MZML. A lot of people also use like MZXML. Those are two um, uh, fairly comprehensive, powerful formats for uh, using these data, and they can basically both take in the raw spectra, but also the process data. And the process data would just be a list of, oh, here's I had a peak with this intensity at this M over Z. Like that would be processed, whereas the raw would just be, just all the counts, all the intensity. Okay, so generally, to do any serious analysis, we'll convert the vendor formats to open formats, and you can go back if you're interested and look at this slide, and it's got some names of software that you can use to do that. And then I want to go through this just so you guys have it, because this is one of the areas where I think there's really room to improve things, and so there's a number of steps in just going from that raw spectrum coming off the machine to something that we would use for later analysis. In particular, the spectra could be noisy, so we might want to correct that. We might have to figure out like what's the zero, what's the baseline by comparing it to blank samples that we know don't have anything in them. Then we have to look at the raw data and identify what a peak is, right? And so that peak, remember, we're watching things as they come through in a time series through our chromatography column. And so it can kind of be confusing if it's noisy, like, is this one peak or is it like two peaks that are sort of on top of each other? And if it's two peaks on top of each other, then we have to figure out how to like estimate what those peaks would look like if they were separated, right? And figure out how much of the intensity is due to molecule 1 versus molecule 2. And then we want to integrate, because we don't really care necessarily how long it takes one particular molecule to get through the chromatography column. So if it gets really spread out and the intensity is low, that's going to mean there's, it's the same amount as if it was really narrow in a narrow band coming through the chromatography column, but the intensity was high. So we want to be able to identify the peaks and integrate under them. And then if we ever want to say anything about this sample besides what's in there, we want to say, is that high or low? Is that more or less than we expected? Is it, which are the molecules that tend to be upregulated versus downregulated in healthy individuals versus, let's say, cancer patients or something like that? We need to be able to take these raw spectra from one run and then align it and figure out like where these peaks in this two-dimensional space of M over Z versus retention time, which peaks correspond to which peaks. Okay, so there's a number of software packages out there. I think CentWave is one of the popular ones, which is fairly good. And this is basically software to identify these peaks. And so I'm not going to go in detail through the algorithm, but it basically uses wavelets to try to identify the peaks. And but I do want to show you sort of like what the raw data looks like. So here on the x-axis, now we're looking at like one window through a chromatography column. Let me move this so we can see. So this looks like about 1 minute worth of chromatography. And you can see each of those dots being a time point, and then the color of the dot tells you like how much intensity there was. And so you can kind of see, like, even within what is probably like a single molecule, we see a little variation in M over Z, and then we also see peaks that are like quite close to each other, but probably distinct. And so you can see what some of the challenges really are. Um, right, I might zip through here, but sort of show you kind of what it's doing. So if you look at the bottom here, here's some more data, and now we're focusing in on like one particular narrow window of M over Z, and we're looking at it over time. And so here you can see what the raw spectrum looks like, and what the software is doing to model it. It's essentially taking these raw data and then identifying 4 different peaks, so 4 different molecules passing through. And then you can see, there's additional peaks like here and here and here. And so we need to draw some threshold for, what we think is actually there versus like what's noise. And of course, then when we compare run to run, generally we're using, we can reuse the same column, but then the retention time of the same molecule in the same column is going to change over time. And so if we're running like hundreds or thousands of experiments, it's really hard to compare the result at the beginning to the result at the end. And I think that's also something that where I don't know if there's algorithms that are taking advantage of the fact that the run from one sample to the next to the next to the next changes very little. So you could imagine like taking that whole series of data and then trying to see how the peaks are gradually moving from point A to point B. I think that's an area for improvement on some of these algorithms. Okay. And then, so the output of these algorithms that analyze the raw data are essentially just the detected features, and getting that good feature list, that's what's really key to reproducible, reliable downstream mass spec data. And so the features are basically going to be the M over Z, retention time, and intensity. And then integrated to the peak area. Okay, so those are the basic features. We want to be able to take some of those features and then identify what metabolites those are. And so there's a few problems with that. First, there's many different molecules for most M over Z ratios. So, if we have a super high resolution machine, so we're not just getting the M over Z, but we actually get the chemical formula, then that helps reduce the number a little bit, but there can still be multiple different molecules that essentially have the same chemical formula, but structurally they're very different, and we'll look at one of those in a minute. And so how do we differentiate between them? Well, that's where fragmentation can help us. So if we're using like a hard ionization method, we may get fragments while we're generating the data itself, while we're ionizing the sample. We might get the full molecular ion and also a bunch of fragments. But if we're using a softer ionization, then we may have to deliberately fragment those ions, and that gets into what's usually called MS-MS or MS2, MS squared, and that's where we, our first pass through the MS is identifying what's the mass of the parent compound, and then we can target specific parent compounds to then fragment, and usually they're fragmented by colliding them with an inert gas or something like that. Okay, so let's look at a simple molecule like butane, and I'll show you some spectra that I think were generated through a hard ionization where we like hit it with an electron beam. But the principle is kind of the same. So we have the parent butane molecule. So we have the CH3, CH2, CH2, CH3. So the molecular weight is basically 58. But we could imagine cleaving any of those bonds, right? And if we cleave those bonds, and in this case, let's say we're cleaving it by hitting it with an electron, we're generating a free radical that has an extra electron and then a positively charged ion on the other side. And so if we're running our mass spec can run in one or two modes. We can look for the positive ions and we look for the negative ions. So we're looking just at the positive ions. Then when this butane is fragmented, we're only seeing that molecule on the left, right? So if we look at the first fragmentation where we just cleave off that CH3, then we've got something with molecular weight 15. If we cleave it in the middle, we get molecular weight 29. Cleave it, so we have three of them in a row, molecular weight 43. And then, in some methodologies, it's more common to get a proton attached to the molecule, and that's how it gets ionized. And so in that case, we get a molecular weight 58 or molecular weight 59. And so here's this is just from like the NIST website. If you look up what the mass spectrum for butane is, this is what you'd see. And so you can see that molecule at 58. This is the molecular ion. It's kind of the parent. It's the butane. This is what's called the base ion. It's the ion for this molecule. Using this technique for this molecule, this is the ion that has the highest intensity. And so we just scale that to be like 100%, and then we measure everything relative to 100%. And then you can see here, all of the different versions of butane fragments that we looked at on the previous slide are here, but there's also additional ones because there can be additional ways to ionize them, or there can be chemistry that happens after the molecule has fragmented. Okay. And certainly for butane, people have gone through and annotated, all of these and exactly where they come from. And then, anything that has carbon, if we were to look really closely, we'd see something up here at 59, because, I think 1 or 2% of every carbon is carbon 13 and not carbon 12. So in a molecule like butane, we've got a 5% chance of seeing everything with a mass number one higher. So even a simple molecule, when it's fragmented, can lead to a fairly complicated pattern. And in general, it's hard to predict what those patterns are, but and the patterns are going to be different depending on how they're fragmented. But there are big databases for many chemicals, especially any chemical from like central metabolism and chemicals that have been studied before, there are large databases of what those fragmentation patterns look like. So here, we can look at butane, which we just looked at. Here's the spectrum, versus methylpropane. Okay, so same chemical formula, but obviously a different molecule because it's got one of the methyl groups branching out of the middle here. And it's gonna generate different fragmentation patterns. If we look, most of the peaks on the top are still peaks on the bottom, and vice versa. It's only the relative intensities that have changed. So, for example, in butane, the parent molecule, the molecular ion here at 58, is higher in abundance than it is in 2-methylpropane. Okay, so 2-methylpropane, more of that molecule is being fragmented, and then we can do things like we can look at the ion at mass 27 versus the ion at mass 29, and for butane they're about equal, but for 2-methylpropane, one is significantly higher than the other. And so we can look at just those small variations in these fragmentation patterns to distinguish molecules that are actually quite similar. Okay, so in any mass spec experiment, there's a number of factors that are important for comparing one to the other. First is the type of column. So there's different columns, and we generally buy them from the same vendor. So if you really want to compare your results to data that you've generated in the past, we'd go back to the same vendor, buy the same type of column. In general, I said we could collect two different types of data. So we could collect either the positive ions or the negative ions. And depending on what we want to do, it may be that one or the other of those is better at capturing the molecules of interest. So back when I was doing more wastewater surveillance, one of the projects we worked on was trying to generate public health data looking at illicit drug use. So we have some records of for prescription drugs, how they're being prescribed and what their sales are from pharmacies and things like that. So we can sort of track the use of prescription drugs. But from a public health perspective, it's also important to understand the use of some of these illicit recreational drugs, and the data sources that we have are very limited, right? If you're using an illegal drug, you're generally trying not to report it to like any central database. So we would look in wastewater, and it allowed us to make some important inferences that were actually contrary to what people believed about illicit drug use. Just by quantifying those drugs in wastewater, and that can be used to kind of improve public health. One of the things that I found interesting doing that is we did everything positive ion mode, because almost none of the illicit drugs generate ions in negative mode. So that was just based on the type of molecules we were studying, that kind of influenced specifically what type of experiment we would run. And so then we would just run positive mode, and we'd reuse the same type of column and things like that, same machine. Type of ionization is going to matter. The type of machine, the mass resolution. So like the FTICR, it gives you crazy resolution, and you can really identify the masses, and then you can use those to make better predictions downstream, but it might not be as comparable with other machines. And then even the fragmentation. So apart from the different methods of fragmentation, even on the same machine, we can have a fragmentation that is data-driven or data-independent. And so if the fragmentation is data-driven, then we're going to get a mass spectrum in our first pass through the mass spec, and it's going to tell us here are all the parent molecules. Here are their different M over Z ratios, and then we can program the machine. Okay, like a common program is pick the biggest one, pick the ion that is most abundant at this point in the chromatography, and then get a secondary fragmentation spectrum from that one. And in general, like we might not, depending on the type of machine, we might not be able to get a secondary fragmentation pattern for all of the peaks. So we have to decide which one to pick. But we can also do data independent, and we can just program in, if here's a peak that we know corresponds to two different molecules that we want to discriminate, and we can say whenever you see a peak like this, then fragment it for the MS2. Okay. I want to take a minute to talk about some really cool work going on at MIT. And so this is from Connor Coley's lab, and it's a program called Iceberg. And what Iceberg does is it's taking these patterns and then identifying what the potential molecules are. And relevant to this class, it's using a lot of the machine learning methods that we've talked about. Oops. So in particular, it's using transformers. It's also using graph neural networks. And it's using a particular type of transformer that we haven't talked about, but I think is pretty interesting and worth thinking about. So essentially how the algorithm works is, we start with a parent molecule. And so we might pick this because we see a particular M over Z ratio. Maybe there's 3 or 4 different molecules in our database that correspond to that M over Z. And so what we want to do is figure out, this spectrum, even if we don't have it in our database to compare to, could we predict what the spectrum looks like? And for our MS2, the spectrum of the different fragments, can we predict what it's going to look like? Then look at the spectrum and compare to our prediction. And if it's really close, then we're going to assign that peak to be the to be that molecule, right? And maybe there's 4 or 5 different molecules like that, and we could pick the pick the best of them. So essentially, what happens is we start with a parent molecule, and then there's a neural net that decides how to generate fragments of that molecule, and you can see that in kind of figure A from the paper here. Like, what are the different ions that are coming out of this parent molecule? And this is essentially an autoregressive neural net, because once we've generated a fragment, like there in the first layer, now we can take that fragment and apply the algorithm again to see how to generate fragments from those fragments that came out of the first step. Okay. And then all of those fragments may, all of those processes might be happening, and so we might see all those different fragments in the final spectrum, right? Because the final spectrum, it's not just one molecule; it's looking at all the different pathways through here. So how do they decide where to fragment things? Well, they're using a graph neural net, and we talked about graph neural nets, especially for molecular graphs that can store really pertinent information on the particular atoms or bonds. I don't actually remember from this paper if the feature vectors were on the atoms or the bonds, maybe the bonds. And that's gonna, be used to try to predict like which ones are getting cleaved. And then once we have a set of likely fragments from that initial model, then what we're going to do is we're going to put those in. So we'll have we're putting into our transformer what looks like a sequence of different fragments, but it's not really a sequence because there's no order to it. But if it was a normal transformer, we'd be seeing like a protein sequence or natural language sequence. This is different because it's all these fragments. There's no particular order. It's more of a set than a sequence, an unordered set. And so there's another type of transformer model called a set transformer that's especially well suited to handling this type of data. So each element in the set has a graph neural net that represents the parent molecule. It has another like feature vector that represents the output of the graph neural net for the particular fragment. And then it has another feature vector, which they call the context vector. And that's going to store information on, probably like the whether we're using positive or negative ion mode and what type of machine it is, and just whatever other context that we want to put in for, for purposes of the machine learning training. And so now we have all these different fragments, and what we want to do is say if these are the possible fragments that we have, how much intensity do we see for each fragment? Because some of them, the intensity might be zero, right? And what we need is for all those different fragments to kind of talk to each other and figure it out. And so set transformers are a lot like regular transformers, but they have a few, probably like two important properties. One is they are invariant to permutations of the input data, right? So protein sequences, you can't permute the order of the amino acids. Like you will not get the same representation out of the model. Natural language, obviously you can't, permute the order of the words in a sentence. But here we do want to make the output and the internal representation invariant to permutation, which is quite easy. We just don't put in the positional embeddings. So if we don't put any positional embeddings in and we run it as a transformer, then we're going to get like a set transformer. Set transformers are cool because they do a little bit more than that, and they can operate on very large sets. So in a transformer, we're comparing everything in some stretch of sequence. We're comparing every single word with every single other word. And maybe we're downweighting it based on how far apart they are, but we're still doing this very large n squared computation. In a set transformer, each fragment in the set has its own unique feature vector, but we're never comparing the different vectors to each other in like an n squared way. What we do instead is there's a smaller set of vectors. Oftentimes people, even for very big sets, people use 10. And they're called, I think, like induced vectors. And so each of the fragments, each of the feature vectors from those fragments will send information into these induced features. And then each of those fragments will then get information from those induced features, right? And so those induced features, in this case, maybe one of the features would be, oh, this fragment includes this big ring structure, right? That would be an obvious feature that might be learned by a model like that. And so then for each particular fragment, it can look at that induced feature vector and say, hey, how many of the other fragments out there have like also include this ring? Because if there's none of them and I have the ring, then I'm going to take up more of the intensity. But if there's a lot of things that are already very high intensity that also have this ring, then like maybe I'm going to be lower intensity, right? So it's just a way for everybody to share information with like a fixed number of sort of salient features for this problem back and forth, so you avoid the n squared computation. Anyway, so that's what set formers are. And these are the data that go into the set transformer, and then from there it can predict these intensities, and it makes a simulated spectrum. And then we compare the simulated spectrum to the actual. So they do this, they compare to like a number of state-of-the-art tools, and they seem to do better on two different data sets of compounds and their spectra. Apart from this tool, which doesn't seem very good, it's the most computational expensive and the least accurate. They seem to be the best performing, but also the computationally most expensive. But this was kind of cool. This is sort of the predicted versus actual spectra for a number of different compounds, and many of them look quite good. Especially this one looks very good. There's many different fragments that you see from this molecule, and the relative intensity and number of peaks is looks quite good. So that's kind of state of the art and cool that it's happening here at MIT. And the last thing I'll mention, and this is like the only thing I'm going to say about it, but it is like an entire lecture unto itself, is using mass spec to look at proteins. There's kind of two ways to do this. We could look at the entire protein, or we can more commonly fragment that protein into smaller peptides, and then compare those to what we expect in the database. So we can either go in trying to annotate a handful of proteins that we really care about, or we can try to discover like what are the proteins that are interesting in the sample and what are the ones that are changing in response to some perturbation. So a few steps. In either case, we're going to extract the protein. We're going to generally digest it. And so we can use different proteases, and we know where those proteases cleave, right? We know the amino acid sequence, where if we're looking at trypsin or something like that, we know where it's going to cleave. So we can look at our, let's say, human genome, build, in silico model of all the proteins, do a trypsin digest, so we know what the peptide fragments after trypsin digestion are going to be, and we can do a few different digests with different proteases, and then we do mass spec on those and compare those to our database, and then try to identify different peptides. So if we see specific peptides there that match what we would see in the database, that might give us, in the best case, we get a peptide and it matches like one protein in our genome, and we're like, this now serves to quantitate this protein. In a more realistic case, we get a peptide and it can match one of a small group of proteins. And so then, then we can look at all the different peptides and we say, oh, actually, when we look at all of the peptides, we see that it probably the like all the data could be explained by this one protein as opposed to some combination of all the proteins in this group. But those are trying to deconvolve those signals is part of the trick in proteomics. And comparing how that changes across samples is important. And then another technique that's very powerful and useful is looking at post-translational modification. So, for example, phosphoproteomics, we can look at what is the phosphorylation state of one particular protein or another. It tells us something about signal transduction. Anyway, that is it. So thank you guys for coming.